Should we start? Okay. So welcome all to our next BioGenome Analysis and Applications seminar. Today we have the honor to host here Toni Gabaldón, the chair of the DAC Comparative Genomics Subcommittee. Toni is a research professor jointly affiliated to the Institute for Research in Biomedicine and the Barcelona Supercomputing Center, where he leads the Comparative Genomics group. Today, he will talk to us on how to detect orthology and paralogy relationships and how to apply the phylomeDB approach to newly sequenced genomes. Toni, the floor is yours.
Thank you, Teresa. So I'm sharing my screen, and hopefully everything will work. You let me know whether you see.
We can see your screen.
You see full screen?
Yes.
Perfect.
Yeah, perfect. Okay. All right. So thank you for the introduction and for giving me the opportunity to talk to you about orthology and paralogy prediction and what is the phylomeDB approach to newly sequenced genomes. So we are living in this era in which we are starting to have access to the genome content of almost every single species that we have access to. And I'm pretty sure this will be realized, at least for some groups of organisms, very soon. This means we will be able to look at this tremendous amount of biodiversity, not just by looking at nice pictures like this one, but also looking at the genetics of each one of these species. And that's very amazing for scientists like us that want to understand biodiversity. But that's also very overwhelming, the amount of information that we are going to have. And we need to organize this data and also have the tools with which to interpret this. So I argue that comparative genomics is an essential tool to understand this biodiversity once we have information of the genomes of each single species. First of all, because it allows us to organize this deluge of data into an evolutionary meaningful way. Like we can classify organisms into different clades and we can look at what are the evolutionary relationships between them. And then on top of these phylogenetic trees, we can start mapping this diversity and observe how this diversity has evolved. So this helps us to organize this, to make, to reconstruct the evolution of the different species, to know when different innovations appeared, but it falls a bit short to understand how this happened. And if we want to understand how these, these different adaptations and these different changes in morphology happened, we need to go deeper. We need to move from a species tree into looking at the genomes of each one of these species. And this is because, each one of these species is not just, a leaf in such a huge tree, but also has a lot of traits that we want to understand why they are different from the others and how they work. And for that, we need to go into the genomes and start looking at the functional elements in these genomes and start comparing. We have no escape from comparing these different genomes. So a key step in our understanding of this biodiversity will be to establish relationships between these functional elements in the different genomes. First, we need to recognize them with genome annotation and know where they are. And secondly, we need to compare them with those of other species. And an important part of these functional elements are the genes. So we need to not only count the genes and know which genes are in each species, but we need to establish relationships between genes in the different species to know which gene is what gene in the other species. And this brings me to the old concept of homology that we use to classify, genes that belong to the same family. So I will go back and remind that homology is a core concept in comparative genomics, but it's a concept that we inherited from comparative anatomy. And in those times when anatomies were comparing the different morphologies and structures of different organisms with those of fossils, homology was coined as to mean the same organ, regardless of their shape and function in different organisms, but the same organ because it descended from a single organ in the common ancestor of these organisms. So in this picture, we see the forelimbs of different animals like orangutan, the dog, the pig, a cow, tapir, and a horse. And they look all very different, and they also have different functionalities for these different animals. It's not the same the hand of an orangutan or the extremity of a horse. And they look totally different. They have different functions, yet they derive from the same forelimb of the common vertebrate ancestor of these organisms. And therefore, they are homologous. So in contrast to compared anatomies, to compare genes, we don't have access to the fossils. Okay, you can say we can sequence now ancient DNA from the Neanderthals and so on, but this is very limited amount of time. In general, we don't have access to the fossils of gene sequences, so we cannot directly compare to ancestral forms for the genes. So we need to rely on sequence comparisons, and that's also a key tool in comparative genomics. We will always have to compare sequences with BLAST or other organisms to reject the null hypothesis that the observed similarity is the result of chance. If the observed similarity is very special, cannot be the result of chance, then we have to assume that these two genes are similar because they have a common ancestry. Therefore, they are homologs. And this is the concept of homologous groups of genes, which are also known as gene families. And gene families or homologous groups of genes are genes that derive from the same ancestral genes. And they can be very intricate. They diversify. Similarly to species, they diversify enormously. But in addition to what diversification occurs in species, they also diversify by duplication, not just by speciation. So they can give rise to very intricate evolutionary relationships. And this is a tree of the kinase family, and only sequences from humans are represented in this tree. So you can imagine how the tree could be if you will take, for instance, all the kinases in all the vertebrates. But that's homology that relates to genes that descend from the same ancestral gene. But we often talk about orthologs and paralogs. And in many ways, we are interested not to stop at the level of homology, but we want to distinguish different types of homologs as orthologs and paralogs. And these words are very important and are used many times, but still, I think there is common misunderstandings of what orthologs and paralogs mean. Maybe not in this group of people that is listening to this seminar, but still in the literature, we still find common misconceptions about orthology and paralogy. So I put you some sentences here. All these sentences are wrong, but I still find them from time to time in scientific publications, like assuming that orthologs are homologous genes that have the same function, also saying orthologs are homologous genes in different species, while paralogs are homologous genes in the same species. This forgets that paralogs can also be present in different species. The ortholog is the most similar sequence among the homologs in another species. That's not always the case, or that's not the only ortholog that one gene may have. Also assuming transitivity, like if A is orthologous to B and B is orthologous to C, it's sometimes assumed that A and C are orthologous. Also using the concept of orthology to refer just to genes that are present in single copy. And also from after duplication, assuming that there is one of the duplicates that will remain being the ortholog, while the other one will turn into different thing. So all these are misconceptions that are still used in the literature, and I think they reflect the fact that sometimes we don't think about these terms. We use them, but sometimes we forget what's the original definition of this. And for that, I will like to remind the original definition by Walter Fitch. He realized that homologs are genes derived from the same ancestral gene, but he realized that there were two fundamentally different ways in which two genes can derive or diverge from a common ancestor. So in one case, the gene will be duplicating, so that for some time, two copies of the same gene will descend side by side in the same organism. And in another case, the genes were not duplicating, were simply in a genome, in a species that then diversifies into different species. And the genes carried by each of these species will be differentiating from their ancestor. So he was calling the first case paralogs, like in parallel, and the second case he was referring to them as orthologous, from ortho exact. So these are the two definitions of orthology and paralogy. And for that, I always like to extend this definition to several corollaries, like things that derive from this definition. And the first thing to remember is that this definition is purely on evolutionary terms. It's not related to function nor synteny in any case. So it's similar to homology. I mean, we may have orthologs that do completely different things, but if they derived from the same gene through speciation, they are still orthologs. Also, that orthology is a pairwise relationship. It's not a group relationship. It's a relationship between two genes. There's no limit in the number of orthologs and paralogs that a given gene can have. There's relationships like many-to-many orthology relationships, and the concept of co-orthology, like one gene is equally orthologous to two other genes in another species, is also important to remind. Also, there's no limit of how ancient or recent these ancestral relationships will be. I mean, we share orthologous genes with bacteria because we share a common ancestor. And also, orthology is non-transitive. And I won't show a specific example here, but just to remember that if you know that A and B are orthologous and B and C are also orthologous, it's not always the case that A and C are as well orthologous. So this is a typical example, the globin gene. Like there was one ancestral globin gene in the vertebrates, and then gene duplication generated the alpha chain and the beta chain. And then in all vertebrates, through diversification, each different species will have an alpha copy and a beta copy of this gene. And we will call all alphas, the alpha chain in the frog is orthologous to the alpha chain in the chicken, and all alpha are paralogous to all beta, meaning that the alpha chain in the frog is, of course, paralogous to the beta chain in the frog, but also paralogous to the beta chain in the mouse. And this is relevant because it also relates to slight functional differences and properties of these two different chains. But that's a simple example, just one duplication followed by speciations. But if you start having duplications and speciations alternating each other and being in occurring several times, you can have very intricate relationships. And this is one schematic tree in which I just showed two species, Homo sapiens and Mus musculus, and then we have a series of duplications happening before or after the speciation of human and mouse. And this leads to very intricate relationships in which we can have one-to-one orthologs, but many-to-many orthologs. And we have also within species paralogs and between species paralogs. So just for this, just to remind that resolving these orthology and paralogy relationships is sometimes a bit tricky. So, but we have already the concept of homology, like genes derived from the same ancestor. So why cannot just use this concept, homologous genes, and why we seek for orthologous genes? Why sometimes it's better to have orthologs than just homologs? Well, there is three cases, I will say, in which it's key to have access to orthology relationships beyond homology relationships. The first one, and one of the most important, is phylogeny. Because they derive through speciation, then only sets of orthologous genes are expected to reflect the species tree or the species divergence. Although I would like to remind that there is some exceptions to this, for instance, horizontal gene transfer, hybridization, etc. But this is one simple example. If we go to the globin gene, and imagine we are interested in reconstructing the species tree, and we take through, only these genes from this branch, that they are all orthologous to each other, we will recover the correct species tree. But if we just care about homology and we simply take one member of the same family from each of the species, we may end up choosing one gene that has a paralogous relationship with the other genes. And this will lead to the wrong tree. Well, I think the tree, I had to change the tree, which I didn't, but it will be mouse in the outgroup, and then the frog and the chicken closer together. So that's one thing to remember, that for phylogeny we always need to use genes that are orthologous to each other, at least when using a gene concatenation approach. But also orthology provides us a much higher resolution when comparing two or more genomes. Imagine we have this relationship in these 3 different species, and we know that there is 2 copies of the gene family in human and in mouse, and only 1 in frog. But this could result from different stories of duplication and speciation, even if we include gene losses. So only by resolving orthology rather than homology relationships, we will know which gene is closer to the others in the other species. And finally, it's important for functional inference, because orthologs, as compared to paralogs, are more likely to share the same function. And I stress this is more likely. It's not guaranteed that orthologs will have the same function, and it's not guaranteed that they cannot diverge in function. In fact, they diverge continuously. But duplication, in a way, promotes functionalization, because subfunctionalization and neofunctionalization are 2 processes in which the retention of gene duplicates is favored. Because after having 1 gene duplication, we initially have a state of genetic redundancy. We have 2 copies of the exact same thing, which, unless dosage is important, usually there's not a particular advantage of having 2 copies of the same gene, and one of the copies will degenerate after accumulating the first mutations, and will become a pseudogene and eventually disappear. This is the fate of most gene duplications during evolution. But if something happens before that in terms of function, like the 2 paralogous genes, they lose different functional properties, and only with the 2 of them you can keep the ancestral ones, and this process is called subfunctionalization, then there is an advantage of keeping the 2 copies, because it's the only way you can keep the same functions. And also, if one of the copies acquires a new function that is useful, then there will be also pressure to keep the 2 paralogous genes. So because subfunctionalization and neofunctionalization promote the retention of duplicates, then duplicates have an additional pressure to change the function as compared to genes that do not duplicate. So if you will agree with me that it's important to predict orthologs rather than just stay at homology, then we will need to look into methods for predicting orthology. And I will not go in deep into the different methods, but basically, probably many of them, and probably you have your favorite one. I will just remind that we can classify them into those that are blast-based, that you only need to do sequence comparisons, and those that are phylogeny-based, in which you need to reconstruct a phylogeny, a gene phylogeny. So in the blast-based approach, these were the first methods that were developed for automatically compute orthology between different genomes. And the first ones were pairwise, like best reciprocal hit, in which you do a blast from one species into the other species, you get the best hit, you do the reciprocal blast, and if the same hit is the same, if the query as the previous blast, then there is a bidirection in this best hit relationship. So these are assumed to be orthologs. So of course, there's one important limitation of this method, is that by definition only provides one-to-one relationships, while we know that there is many-to-many and one-to-many relationships. To solve these methods, like here in C, like in Paranoid, were developed to alleviate this problem, and they combine the base bidirectional hit finding with a blast in the same genome and detection of imparalogs. More sophisticated methods are those that build networks. They do all against all comparisons in a set of genomes, and they build a network of similarity relationships. And then from these networks, clusters of genes are selected, and these are called orthologous groups. And this, I will say, is the most popular method nowadays, and methods like OrthoFinder and many ECNOC and many others are based on this approach. There are also other methods that require constructing phylogenies, gene phylogenies, and I will classify these methods into two main groups: reconciliation methods, in which you know a species tree or you infer a species tree from the data, and then you reconcile the gene tree and the species tree so that they agree to each other by introducing duplications and gene losses. And sometimes also some models allow to also contemplate for horizontal gene transfer. So by doing this, you do this reconciliation, and you end up knowing which nodes had a duplication and which nodes are an speciation. A simpler method is the species overlap method, which is the one we used in phylomeDB, which simply traverses the tree and imputes a duplication at every node in which the two descendant branches or clades have one overlap in the species. So if we see the same species here and the same species there, then we have evidence that a duplication occurred here. And the rest assumes is only speciations. So there are also combinations between these different methods in which you can first start making a family, like an ortho group, by using these network-based methods, and then for each of these families you can reconstruct a gene tree and detect duplications and speciations events. So I won't explain more about these methodologies, but I will recommend you to check the Quest for Orthologs website. The Quest for Orthologs is one consortium of many different groups that work in developing orthology prediction methods, and also communities that require these methodologies. And there is a meeting every 2 years. This year is in July in Montreal. So I invite you all to check this website and check also for the meeting. And the website maintains a list of many different databases and methods to predict orthology. So, but here I'm going to talk to you about the phylomeDB approach, which is our own approach to orthology, but also to many other things. I will say that phylomeDB is not just an orthology prediction approach, but more an data mining approach for gene phylogenies. And first, I would like to kind of provide some arguments on why I prefer trees over the orthologous group network-based definition. And this is one aspect that we have encountered many times when looking at different gene families and using also these methods. We are also use this type of network-based methods for some projects, but they have the issue that okay, you can build a network of similarities between all the genes in the genomes of interest, and then the difficulty is how you derive from this network defined clusters of orthologous groups. And there is always one parameter that will be how stringent, what's the level of granularity that you want to define this into families. So you can be not very stringent and get everything that has a hit into the same family, and then you have very large families, and maybe you will include proteins that share only one domain, or that are not, or you are fusing maybe two different families because there's one error in annotation in one of them. Or you can be more strict, more stringent, and discard those genes that are, more collateral, not really connected to all of the rest. And you can be even more strict and only keep those proteins that are truly very well connected between them. but there is, there's always a balance between, losing true positives or getting false positives into your groups. And it's very difficult to establish this at the level of stringency at the genome-wide level, because each family has a different story and each family has a different type of network, and it's very difficult to fit to choose one parameter that fits all families. So for some families will go fine, some families will go wrong. And I mean, this is one example we have studied in more detail, like how many families how many orthologous groups you will see here. Some people will answer there is just one, but some methods do not cluster all these proteins into the same family. Sometimes they divide into three, like it could be the 3 vertebrate group, but maybe sometimes one of these vertebrates will get some non-vertebrate species. Sometimes P73L and P73 go together because they diverge much less, and P53 is considered a totally different family. So different methods will do different things with this one family. So also orthologous groups are useful, easy to use and fast, but they work at the level of a group, despite their name that is called orthologous group, it's important to remember that not all the members within an orthologous group are orthologous to each other, because the orthologous groups include imparalogs, like all the genes derived from duplications after the common ancestor of this family will be included. It's difficult to determine the granularity, as I explained, and I think in essence it's a simplified representation of the evolution of gene families, because it's kind of reducing the complexity into 2 dimensions from something that is actually more difficult. So the way I look into that is that, yeah, a tree tells you most or the whole story, like what are the specific relationships between each pair of genes. You also can visualize the distances, and I will consider, okay, this could be like a 3D representation of the evolutionary history of a gene family. And making a group of that is kind of reducing this complexity, and it's maybe easier to use, easier to compute, but I think you lose part of the information. So based on that concept, the phylomeDB approach is kind of trying to use the gene tree as it is. And in essence, is doing what a phylogeneticist will do to understand the evolution of a gene. Like if you will be interested in knowing the evolution of a gene, probably you won't do this network reconstruction. You will start blasting, selecting homologs, aligning those, and reconstructing a phylogeny and looking at the phylogeny. So the phylomeDB approach is doing exactly that, but repeating this in an automated process for every gene in a genome of interest. And this collection of gene trees for every gene in a genome of interest we call a phylum. And then the phylomeDB approach is not only to reconstruct that, but also to mine this phylum to address the questions of interest. And over the years, we have developed and improved one pipeline that is evolving, that basically goes from, from the genes in your genome of interest to doing comparisons with databases and selecting the homologs of interest, making multiple sequence alignment, and then making maximum likelihood trees. So we generally use tools derived by others, such as now we use IQ3 to make the phylogenies, and MAFT, muscle, and other methods for the alignments. But in this time, we have developed also some tools in the group, like Trimal, I think has become quite famous now, and many people is using it. We derived it because we wanted a trimming algorithm that was first looking at the alignment before deciding what parameters to use for each family. Also, we developed ET, which is a tool to interact with all these collections of gene trees. So this is continuously being developed in Jaime Huerta's lab nowadays. And then also phylomeDB, which is a database to include all these phylogenies that we reconstruct. So just to show one example of the care we take in this pipeline is like, for instance, for the alignment, we consider the alignment is very central for what you do afterwards. So we don't trust everything to one aligner. We use 3 different aligners, and we align the sequences in forward and reverse. And you may think that, okay, you're gonna get the same alignment, but not. Try it, and you'll see that if you align the same sequences in reverse, you'll get quite a different alignment. And this is because there's heuristics in the alignment process that sometimes introduce a gap always to the right or to the left. And when doing this, you will change that. So then we have 6 alignments, and we build a consensus from these 6 alignments, getting the pair of residues that is most often seen among these 6 alignments. And that's interesting, not only because we think the consensus alignment will be better than each of the individual alignments, but also because it provides us information of which regions of the alignment are conserved, like not conserved, but stable, and they do not depend on the algorithm, and which regions are variable and will change if you change the direction or you change the algorithm. And we use this to trim with trim all. We trim the regions that are very unstable. So we keep regions that are more robust to the alignment procedure. So all these phylogenies, they end up in this phylomeDB database, which is now in version 5, and it has several millions gene trees. It's the largest repository of gene trees nowadays. And there in this database, the users can interact with every single gene tree. So you can browse the trees and manipulate the trees. We also map domains into the different proteins. And of course, we predict duplications and speciations and derive orthologs and paralogs based on these trees. So, but we don't do this just for the sake of computing many gene trees. We do this because gene trees, as I explained you, they have a lot of information. They contain a lot of valuable information that we would like to mine to understand, how a particular trait emerged in one species, or how 2 species diversified from each other. So we can mine these gene trees, and we have the algorithms to do that, to find, for instance, families that show a particular topology. For instance, if there is a discussion between different phylogenetic hypotheses, or if you want to understand whether there is incomplete lineage sorting or hybridization. You can detect and date duplication events. You can look for genes that have accelerated evolutionary rates in a particular lineage. You can find positive or relaxed selection. You can look for families expanded in a specific lineage. You can look for footprints of horizontal gene transfer, lineage sorting, gene conversion, and other evolutionary processes. You can search for genes that co-evolved and also use to predict functional properties. And of course, as I explained you before, use it to predict orthology and paralogy in the most, high resolution way. So just to show, we have been using this to mine existing genomes for different questions that we have had in the past. But today, I'm also want to explain you that this approach can be used for a newly sequenced genome. And the first time we did that was with the sequencing of the genome of the P aphid. So quite a long time ago, but the concepts are still valid for newly sequenced genomes. So we were asked by this consortium to join the consortium to help them into the comparative genomics approaches of this newly sequenced genome. And we proposed to use our phylomeDB approach. So at that time, there were not so many genomes available, and what we did was to compare this aphid genome that had around 34,000 genes annotated to all other sequence arthropods, and also a few outgroups that were this is what everything that was available at that time. At that time was very easy to select which species to compare because you just took all of them. But now this has changed. So what we did was to reconstruct the phylum of this P aphid in the context of all these other arthropods and outgroup species. So in the end, we had a gene tree for every gene in the APSOM. So one of the first things you do is to reconstruct a species tree, And for that, we use a gene concatenation approach, because we had the phylum, we could select genes that are single copy in all the species, and they are present in all the species. These were like 197 proteins that we concatenated and reproduced this phylogeny. So you can see here Apisum, and one of the results is that Apisum is had quite an enlarged genome, and quite a lot of the colors indicate which genes are single copy and multiple copy in relationship and conserved in the different clades that are represented here. So there were two observations. One is that it has quite an enlarged genome, and also the presence of many duplicate genes. So one first thing you want to do with your genome is to annotate, not just find what genes are there, but also annotate what's the function of the genes. And in this case, Apisum was quite far away from the model species. So the model species in insects is Drosophila melanogaster, which is quite far away. And it was difficult to detect, one-to-one orthologs or one-to-many orthologs between these species. We used the trees, and we could transfer functional annotations like Go terms from Drosophila genes, but we could have different sets of rules to transfer these. For instance, if you have a one-to-one relationship, and that is more confident than if you have a one-to-many or many-to-many or many-to-one relationship. But still, you can annotate this to a certain level. And also another useful thing we could derive is that you can also map all annotated functions for all the genes in your tree. And for instance, in this case, you know that the 3 Go terms that are annotated in Drosophila melanogaster, they are also annotated in 2 of the outgroups, suggesting this function is conserved across the family, and making you more confident, because this is one-to-one and the function is conserved, to get this functional annotation. So that's something, that level of confidence you cannot get with methods based on, for instance, just BLAST. So, but yeah, that's one thing you can do, orthology prediction and transferring of functional annotation, but you can do many other things. And in this case, because of the large amount of paralogous proteins, we were very much interested into gene family expansion. So this is a picture showing like families that in paralogs, like lineage-specific duplications in the P aphid, and many of them had 2 members, but 3, 4, and so on. But we had also several families that expanded like crazy, like have more than 40 gene copies that duplicated specifically in the aphid lineage. So this is one example. You have a gene which is single copy in all the other species, and then it goes crazy, bump, in the Apisum lineage starts duplicating, probably at very different times, because you have quite long branch lengths here, but this family duplicated. So how you can interpret this is difficult to interpret just from the tree, but the tree is telling you that there's been a selective pressure to increase the size of this gene family. And this family, by looking at the function, is a transporter, is an acetyl-CoA transporter. So the interpretation, the hypothesis we derived at the time was that because aphids feed on a very specific diet, which is the plant sap, and which is very rich in sugars, but very poor in any other nutrients like amino acids or vitamins or anything, then it's very likely that you really need highly specific transporters to get the most of this nutrient source. And so probably there is a trend or a need to diversify transporters and making them more specific to each of the nutrients you want to capture. That's a hypothesis. The phylomeDB approach is not gonna tell you the total answer, but it's just deriving a hypothesis that then can be proved experimentally. That's another crazy example. This was the largest expansion we found. And you see that there is many genes also in the Apisum, and there's a single copy in all the other species except for humans, where it's 2 copies. And that protein was annotated in Drosophila melanogaster as the Kelch protein, which is annotated as being involved in the ovary morphology. So what relationship could this have two P aphids. So one hypothesis we drive in this case was the aphids. They also have a complex reproductive cycle, and they have 6 sexual females and asexual females that are, viviparous and oviparous females, and they have a totally different morphology of the ovaries. And the hypothesis here was that maybe this diversification helped into shaping these different ovary morphologies in the different females. And some collaborators there, what they did is like looking at the expression of these genes, they found that some paralogue genes were expressed in one type and others in the other. So maybe that could be one factor helping in the development of this unique trait. But you can go beyond just topologies. All this that I explained was based just on the topology, but you have all the information in a gene tree. You have the information of every branch length, and you can go deeper and start looking at the rates of evolution within each branch in the tree. And this is what we did years after, when there were more aphid genome sequence in this other article, where we focused on how duplications in the aphid were diversifying by gene expression differences and functional differences between the genes. So you can go, because you have all the trees, you can make this test of selection for every branch of interest in the tree. So that's one example with the P aphid. Since then, we have been collaborating in many other projects. One big one was this large-scale project with 48 bird species. And there, one of the main questions was resolving the phylogeny, and we were also looking into the differences between species trees and gene tree discordance in the diversification of the birds. In other projects, we have teamed up with groups working on domesticated species, mostly with plants. And the questions there was, the role of crop or the impact of domestication in the genomes, and also the role of hybridization. I'm finishing. So then duplication followed by tissue specialization. It has been a question that we have addressed in these other projects, in the P aphid, but also in turbot and in the bean, the common bean. Other projects are more related to conservation, and there the focus has been different, maybe looking at the signatures of genetic drift, looking for pseudogenes and deleterious mutations, also for signatures of introgression in these species. And some other projects, massive gene expansions, like in the case of the aphid, has been the main question. Also, we have identified whole genome duplications in different organisms. These are signatures you can also detect in these phylums. And overall, in each, I will say that in each of the projects, there's been different biological questions, but the same similar tools, and always starting from the reconstruction of the collection of all evolutionary histories of all genes in the genome. So I think the phylums provide the best picture of the evolution of these species at the level of each individual gene. So many of you in ERGA are now in the process of getting new genomes, and so I'm going to propose you to, if you got a genome, you can get a phylum. And to make things easier for you, we have opened this ERGA Phylums initiative within phylomeDB, which is kind of a way to engage with the ERGA community, in which we have opened a call for collaborative projects, in which you can send us a proposal, like if you have a genome, an ERGA quality level annotated, assembled and annotated genome, and you have some questions, you can approach us, and we can maybe team up and build a phylum for your species and help you address some of the questions. So if you want more information, you can go to the website. So the idea is that if you have an ERGA quality assembly and you have a question that you think can be addressed by looking at the evolution of all the genes, so in essence should be kind of questions that can be addressed with an evolutionary approach, we can meet together and co-design this phylum, decide which species to compare, what will be the focus questions, what's the scope of this phylum. We will reconstruct the phylum. We are based on the Barcelona Supercomputing Center, so we'll use the supercomputer to build this gene phylogenies. And in the end, hopefully, we can address some of these questions that will end up into a publication or some science out of this, but also it will be at the creation of a community resource, because this phylum, I mean, users of phylomeDB and of researchers interested in that species will have the phylum available in phylomeDB, all the orthology and paralogy calls, and also all the gene trees to explore and keep asking other questions. So with that, yeah, I'll just point you to go to the phylomeDB website and get more information. And I would like to thank you for listening, people in my group, especially those involved in the current version of phylomeDB, and also to all collaborators that have been working with us. And with that, I will finish here, and I'm open to questions. I'll stop sharing. Yes.
Thank you so much, Toni, for this amazing talk on this very nice topic. We're all looking forward to use now the phylomeDB. So we have questions, and we have a bit less than 15 minutes, so I will start. So Robert Waterhouse asks, the further back in time we go, the fewer single copy orthologs we can identify. So what species tree methods are possible with so few marker genes, or how to get more marker genes?
Yes, so that's, I think is, so genomes are finite, no? And the more genomes you have, the issue of duplications or losses in specific genomes will always be there. So we have different approaches for that. For instance, is you have to be a bit more relaxed. So even if you have duplications, you can still get one copy for every species and keep the request that each copy is orthologous to all the other ones by carefully looking at the gene tree. Because you can, for instance, for a species-specific duplication, you can just take one of the two, and that will be orthologous to all the rest. It gets more complex when you start having deeper duplications, but we have applied this approach for plants that have many duplications and managed to do this. Then there's this, then there's this other family of reconciliation algorithms like Astral and LRAX that look at all the gene trees and try to build a species tree that is most compatible with most of the gene trees by minimizing the number of duplications, losses, and transfers. And this looks at all the gene trees, including those that have duplications. So I think there is room also to progress in these approaches. So the gene concatenation is true that it has the limits, and especially when you want to have a tree with many species that are very divergent. So I think it's not a good approach for that. It's a good approach when you have a focused question with a limited number of species you want to look at.
Okay. So Christian asks, which are the methodological limitations in resolving these relationships, ortholog and paralog, in divergent species? Does a high-quality genome annotation help in this process?
Of course, it helps. It's not the panacea, because there are problems that do not depend on the annotation. But in this project, we have found many issues that relate on the annotation of the genome. Like, for instance, fused genes. Like if you have two genes together, this will mess up with all these clustering algorithms, and yeah, that's a big problem. If the genes are incomplete, you miss exons, and you have different exons in the different annotations in the different species, you will have issues. There's been a paper, like I think 1 year ago, that shows that if you compare genomes annotated with different methods or by different sources, like one from Ensemble, another one from JGI, another one from NCBI, you'll find more orphan genes than if you systematically annotate with the same approach all the genomes. And this is, noise created also by the gene annotation. But as I said, this is not the only problem. For annotation is one important aspect, but if the species you are comparing are very distant, you have lack of signal sometimes to reconstruct an accurate gene phylogeny and to establish accurately orthology. Yeah.
Okay, so I will not say this name correctly. I'm sorry for that. Vivek Shurans says, wouldn't one orthologs form reciprocally monophyletic groups, ideally? Would that not help to classifying genes? How does one find the underlying reason for deviation from monophyly?
I don't think I get the question properly. So the reciprocally monophyletic is...
So I guess clear orthologous groups where you have one ortholog.
Yes.
That's what I understand. Would that not help for classifying genes? I guess it does.
Yeah, I mean, classifying genes, yeah, the question of gene classification is also an issue. I think in the end, like making orthologous group is a way of classifying genes into a family. The thing is that how I see it, the families they have a hierarchical structure. So you can always subclassify like a subfamily into subfamilies, and depending on the focus, the optimal granularity is different. So therefore, as biologists, I think we are used to have, classifications that need to be relaxed or at different degrees, no? It's like a taxonomy. So sometimes so for genes is the same. We need like a gene family taxonomy to know at what level our question needs to be placed. Yeah.
So Robert Waterhouse asks again, how is the phylomeDB annotation of speciation and duplication nodes on gene trees different from the methods used by Ensemble? And how do they compare for accuracy, etc.?
Yeah, so it's different. I mean, we use a species overlap approach, which is independent of the species tree knowledge. And this is because we use many groups for which we don't have a species tree. And Ensemble, they use kind of a reconciliation approach that if you have a low bootstrap support, for instance, they rearrange the tree into how the species tree looks like. And they try to minimize, I mean, the species tree is done in a way that minimizes the number of duplications. So they take some stand into a species tree expectation. In phylomeDB, we are agnostic to the species tree. Our species tree is kind of a star-like species tree. So we do this because many of the questions we ask are about the gene tree species tree discordance. So if we reconciliate, we lose that signal. So we want to look at the signal, and then we will determine what we want to do with that. No, but we want to detect the signal, and thanks to that, we have detected hybridizations, incompleteness sorting, and things like that.
Okay. I have a question that fits very well after this question. So maybe I can ask it now before I move on to the next two. And that is what happens when you have genes which are very conserved and very short, and where you don't have lots of phylogenetic signal.
So very conserved and very, very short. Yeah, I mean, in that case, imagine like a tree won't make any difference, no? Like you will need to use, in that case, the species tree as a first, inference of how they diverged, assuming there was no horizontal gene transfer, no hybridization. But yeah, that's an issue. Usually the problem is the opposite, no? Because if the gene is exactly the same sequence, there's no variation, at least the function, I mean, you're not worried that they have different functions, and you can clearly establish which gene is what gene in the other species. So usually we deal with the other type of problem, like when you cannot recognize precisely the evolution of the genes. Yeah.
Okay. Then Robert Waterhouse asks, he says that the latest official annotation for the aphid, the apisum, has 20,000 genes, so 16,000 fewer. Were the results replicated with the newer dataset?
I don't know if it's the last dataset we replicated, but we did a phylomeDB version 2, a phylum of the aphid version 2 when some of these issues were solved. And in fact, the phylum analysis helped to identify some of these. For instance, we detected that many of the massive duplications, when we look at the annotation, they were likely transposons. And they were transposons that escaped the masking of the genome because they were families that were not in the databases. And because at that time, the p-aphid was very divergent as compared to any of the other species, then it had many of these. So the phylomeDB approach, and it could be the case for these ERGA genomes, that allows to identify some of these problems, no, like an unrecognized transposon. So many of these were transposable elements that were removed. Other problems were due to heterozygosity and not being, I mean, different alleles not being collapsed in the genome and so on, and this changed some of the results. But these families that I show, they stay there because, as you can see, the copies are very different one from each other. And these are duplications that happened along the evolution of aphids. And we have replicated this with new aphid genomes.
Okay. So Chiara Bortolucci asks, can you expand on the application of your approach in conservation genomics? What was the major objective?
So in the case of course, conservation genomics is something for which you need more population genomics, let's say, than comparative genomics. But comparative genomics can help you to recognize things that happened since that species diversified from other species, and that was the focus on the links. We wanted first to recognize, for instance, what genes that were conserved in other species were accumulating mutations in the Iberian links more often than expected, which changes could be affecting the function. So this was mainly the focus. And in the case of the jaguar and in felids in general, our contribution was also towards finding signatures for past high introgression between different lineages, and that may have implications also for conservation. No? If you think that maybe the hope of one species is maybe putting some genetic material from a close species that is, healthier in terms of conservation analysis. Yeah, but the focus is totally different. Yes.
And there's a last question in 3 more minutes, so I'll be fast. Do you also have, from Vivek Surans again, do you also have coalescence approaches like Astral implemented in your pipeline in addition to concatenation-based approaches?
So not as yet, but we are working into these because we didn't have those because of the large computational demands. So we don't do this for default, but we are using those for specific projects, and we could do this for specific projects. But implementing it for the whole database is probably, not within our reach because of the computational demand. But for some projects, if that's necessary, we can have it.
Okay, can I take advantage of the one last minute to ask you on computational demands that all this has? Because we have so many new genomes coming. What happens with every new version you have with so many new genomes? How do you plan to deal with that?
Yeah, so that's a big issue that we are currently also discussing how to address in the future, because we are not Ensemble, we are not Uniprot, or we are just a group with access to a big cluster. Okay, but this maintenance and updating cost a lot of time. So we want to think on phylomeDB as an incremental project that we don't need to recompute everything. Also for green computing concerns. No, we don't want to burn CO2 just for, but that's an issue because even well annotated genomes, every new version will change a lot of the results. And we have, the quest for orthologs, we have seen that. If we don't use the last version, we miss many of the genes that are, modified or something. So that's something we are currently discussing. I think we need to discuss not only among us, but also with the community of users, what the users will need and so on. But one thing is clear, we cannot do all for all and repeat everything every 6 months. That's impossible.
Yeah, that makes sense. Okay, so thank you very much, Toni. It was a really nice seminar and discussion. And
Thank you.
thank you all for watching. And we have now the DAC community meeting. 5 minutes.
Yes.